I would like to introduce the first speaker of the second session, Professor Kanta Subbarao, with the theme of leadership as a clinician-scientist. Professor Subbarao is the director of the WHO Collaborating Centre for Research and Reference and Research on Influenza at the Doherty Institute. She has worked on molecular virology and vaccine development for emerging viruses that pose a potential pandemic threat, including influenza viruses, SARS and MERS, and now SARS-CoV-2, coronaviruses, in a career spanning over 30 years. Previously, she was chief of the molecular genetics section of the influenza branch at the Centers for Disease Control and Prevention in Atlanta, and the chief of the emerging respiratory viruses section of the Laboratory of Infectious Diseases of the National Institute of Allergy and Infectious Diseases in the USA. Professor Subbarao is an internationally recognized leader in the field of influenza, having published 234 peer-reviewed publications in leading international biomedical journals and 92 reviews, chapters, or commentaries. Please join me in welcoming Professor Kanta Subbarao.
Thank you very much for the invitation to speak here today and for all of you being here on a Saturday. So I'm going to tell you a little bit about myself and my journey. I think we all get to being physician scientists in different ways, following different paths and different opportunities. And as you heard, I've spent most of my career in the US, moved here just under 7 years ago. And of course, 3 of those 7 years were spent, a good part of it was spent locked down in Australia. So at the end of it, I said, get me off this island. So I started off in medical school in India at a medical school called Christian Medical College, or CMC Vellore. It was an outstanding medical institution in terms of the clinical training we got. I was 15 when I went there. My parents were not happy or supportive of my going into medicine because I grew up on an academic institution campus, but it was scientists and engineers. So my parents said, where did you even come up with this? Who studies medicine? But anyway, so I went to medical school. When I finished a few years later, I assumed that I would have a career in clinical medicine. But my life has been a series of forks in the road, and you take one path or the other. So when I was a medical student, I really thought that I really loved ophthalmology, and I thought that's what I wanted to do. And my second choice was pediatrics. And so I applied to residency programs, got into pediatrics, didn't get into ophthalmology, thought, well, let me try pediatrics. If I don't like it after a year, I can always try again. So I went into pediatrics, and I can tell you taking care of kids is a whole lot easier than taking care of adults. And so I did pediatrics, realized that I really liked taking care of the sick kids. And so I went into infectious diseases. So I did, in the US, pediatrics is a primary residency. So 3 years of pediatrics followed by 3 years of pediatric ID. And in that fellowship program, it was very, very well designed. We did all our clinical work in the first year. And in the next 2 years, you had lab time. And it was uninterrupted lab. So you weren't running off and taking care of patients in between. So it gave you enough time to actually learn to pipette and how things are done in the lab. And I have to stress that, Claire Scott said the same thing. I think you really need to give yourself time to learn and enjoy laboratory research. So when I finished my fellowship, I expected to do clinical medicine and combine it with research. And I took my first job. And the way it broke out, I was going to do 5 months of attending on the Peds ID service, and the rest of the time was research. And this was the very late 80s, and I realized very quickly that I needed more research training. So I went to the National Institutes of Health to do more research training. And I went there saying I would do 3 years. And this was my mentor, Brian Murphy, at the time. We're still friends. And when I got there, I said 3 years. I ended up staying 5.5. And absolutely loved it. I went there thinking, I'm going to go find a clinic. I'll find the peds department and do some ID. But by the time I actually got immersed in the lab work, I didn't miss it very much anymore. and I couldn't see taking the time to do it. Other people do it differently. My husband is a clinician-scientist. He followed, we went along the same paths, but he always did clinical medicine as well. So when I got to the lab at the NIH, I really wanted to work on respiratory syncytial virus. I'm a pediatrician. That's the biggest pathogen viral pathogen at a young age. But all those positions were committed, and Brian said he really wanted a physician to work on flu. And I thought, oh man, influenza. Is it even that important in kids? At that time, none of the burden of disease studies had been done. And there's so much literature in flu, and there's all the subtypes. And, I would walk around muttering H1N1, H2N2, N2, just to get it right in my head. And when I got there, I really didn't know any molecular biology. I didn't know how to design a primer. I didn't know how to do PCR. I didn't know how to do anything. And so Brian was very, very patient. And the way they designed the lab at the NIH, and this is a very famous lab. It's a lab. The lab chief was Bob Chanak, who discovered RSV, many of the mycoplasmas, adenoviruses, and parainfluenza viruses. So it's a lab with a very rich tradition in respiratory viruses. And what they did was they paired on any given virus. So not on a given project, but on a virus, they would have PhD postdocs and MD postdocs working together. So there was a real recognition of the importance of translational medicine and translational science. So I, again, wasn't sure whether I would be doing influenza or RSV, ended up working on influenza, best decision of my life. The virus doesn't die as soon as you thaw it and put it on the bench. Influenza genetics are fantastic. It's a segmented virus. You can reassort it. You can mix and match. It's just magical. So I have absolutely no regrets about working on flu. And then when I finished my 5.5 years, I again hit this crossroads. I first took a job at McGill at the Montreal Children's Hospital, meaning to combine pediatric ID with research. For a number of reasons, it just didn't work out. So we went back to the US and I went to the CDC. And I went to the CDC, to the influenza branch, to run the molecular genetics section. And our boss said, we need to do a drill. Someday we're going to get an unknown virus. So we should just do a drill and say, here's an unknown virus, work it up. And she went off on vacation. She went to a dude ranch in Wyoming. She was very hard to reach. And we got a call from Hong Kong saying, what about that virus we sent you? And we said, huh, what? So we dug through our freezers, and there was there was a virus that this very, very astute pathologist from Hong Kong had sent us, and she said, it came from a 3-year-old who died, and I can't subtype it. It's influenza A, but I can't subtype it. So we immediately got it out, and we started doing what we needed to, and I realized, as head of molecular, that we didn't have primers to characterize any avian viruses. It was very siloed. If you worked on human flu or you worked on animal. So I called some colleagues that lived about an hour away, and we met in the parking lot of a guitar store, and he gave me an esky with primers and positive controls and so on, and I rushed back to the lab. So we started working this up, and it was the first H5N1 bird flu virus from a kid in Hong Kong. So, and the group that I led, I was very new there. I'd only been there 3 months. And they said, well, this can't be real. This must be that drill. And I said, no, it's real. And they said, no, Nancy Cox would not have stayed on vacation if this was real. And we said, no, let's pay attention to this. So that was the first case of H5N1. A few months later, there were 18 more cases. And we went through trying to prove that it actually came from the child. So we got the original clinical material, spun it down, stained some cells. The child was dead, so there was no question of doing more investigations in the child. But they did an epi investigation of the daycare facility where there was a little nature strip where they had chickens and ducks, and the baby chicks had all died. And there was, by that time, when we started talking to the veterinary authorities, there was actually an outbreak of H5, highly pathogenic in poultry, that the human infectious disease world didn't even know about. So that's another lesson. We've learned that we need to connect more. So anyway, so that event in the late 90s in Atlanta really again changed my career to working not so much on human influenza, but on avian and pandemic influenza. So I went to the NIH to start a new program. And the program was to develop vaccines against potential pandemic strains of flu. And I worked out there for 14 years. We had a program going all the way from figuring out which subtype to target, designing the vaccines, doing the preclinical studies in my lab, and then we collaborated with the company that then manufactured the vaccines. They gave it back to us, and we did the phase 1 clinical trials. So we did a whole bunch of these things. But the other thing we did, and I'll say that what has been an important part of my research career all along, is that I continue to ask very translational questions. So in 2009, there was another influenza pandemic. So this was the H1N1 pandemic of 2009, which some of you won't remember, especially after the blur of the last 3 years of COVID. But the prevailing wisdom in influenza is that influenza viruses use sialic acid as their receptors. And the sialic acid receptors have a terminally linked to oligosaccharides. And there's a specificity. So all influenza viruses use sialic acid. Human influenza viruses bind sialic acids with the 2,6 lineage linked oligosaccharide, and avian influenza viruses bind 2,3. So dogma develops very quickly in medicine and science. And so the dogma in the field was that avian viruses bound 2,3, human viruses bound 2,6, and that was why bird viruses didn't normally infect us. So we started with the 2009 virus, which had 2,6 binding. And in order to study this a little more, we thought, well, instead of the very controversial dual use, where you put in something to make something worse, we said, okay, here's a virus that came from an animal, spread, clearly had the ability to spread in people. Let's take away that 2,6 and make it 2,3 binding. And if we did that, would we lose transmissibility? So that was the research question. And so we use a ferret model to study. And so we can put ferrets, these are the cages. I don't know, is there a pointer on this? Oh, okay, yeah, that's good, thank you. Yeah, so these are ferret cages. There are 6 cages. Air comes in from the side, it goes through the cage, and then gets HEPA filtered out, okay? So you can put a ferret into one half of the cage. You can separate the ferret by a perforated divider, so the air goes through. You can put a naive ferret next door. Ferrets, if you don't separate them, curl up together. They're very social animals. So by separating them with this perforated cage, divider, they don't contact each other. So only the air from the infected ferret coughing and sneezing. So ferrets cough and sneeze with flu. So they cough and sneeze, air gets transmitted, and then you can look at the naive ferret that you put in there and say, did they get infected or not? So when we did this experiment, the yellows are the infected, and the blues are the airborne contact ferrets. We predicted that the original virus, which is the 2,6 binder, would transmit, and it did, 3 out of 4 cases. But we predicted that the virus in which we changed that receptor specificity should no longer transmit. And to our surprise, it transmitted. So we thought, what's this about? So, but there was a bit of a delay in transmission. So where that arrow is. So it's a couple days later. So we thought, well, maybe the virus is changing. So we then deep sequenced, and this was way back in terms of deep sequencing time. But these are, the blue indicate the nucleotides changes that we engineered. The orange is what was in the original virus. So we took a look at our 2,3 inoculum, and it had all the mutations we put in it. And then we looked at the recipient animal's virus after that got infected, when we didn't think it should. And one of those residues had flipped back to the original wild-type virus. So we said, when did that happen? So we looked at serial time points, and this one residue gradually increased in proportion. So we did a bunch of studies and showed that, in fact, the virus didn't lose 2,3 binding, it gained 2,6 binding, all because of this residue. And so we said, well, why did that happen, and where did it happen? Where in the animal did it happen? So I sent off my postdoc and, and the technician she worked with to try to do this experiment. And what they came back with was, I told them to go and dissect everything they could see, all the tissues separately. And one particular piece of tissue had the revertant. And I said, what was this? And my postdoc, who didn't know any anatomy, said, oh, it was that piece of tissue in the back, so it was soft palate. And we historically have never looked at the soft palate. No one in our field has. So we wrote it up. You see that we finally got it into Nature. When we wrote to Nature with the story, they said, well, this is just a ferret phenomenon. So we said, well, okay, maybe not. And so I told my postdoc that she had just interviewed for jobs, and she had met some people that work with pigs, and pigs are a great model for flu. And I said, okay, your job is to go get pig soft palates. And I said, my job will be to get human soft palates. So I called ENT surgeons who said, yeah, that's operational, it's waste in theater. Yes, we can give it to you, but you need to write the ethics. And then I contact my college roommate, my medical school roommate is an anatomy professor. So I called her and said, hey, I don't need tissue from a living donor. Do you have soft tissue do you have a soft palate from a cadaver? And she said, I can put you in touch with somebody in New York. So I contacted the guy in New York. He said, you're across state lines. We were in DC. And he said, you're across state lines. Why don't you try Maryland? So I called Maryland, and they said, how many do you need and when do you want them? And will you pay the courier? And I said, yes, I'll pay the courier. So I filled out my paperwork, paid the courier, and the day, and I said specifically, I just want soft palate. So this big box arrives, and it's really heavy. And it was a whole head. Yeah, so we had 2 heads. So my postdoc is, of course, this is her worst nightmare. She's not going anywhere near the box. But I had a veterinary pathologist who was doing his PhD with me, and I said, okay, you and I will do this. So we go in there, and of course, you can't even separate the jaw to open up the mouth, and so he had to go to Path and get a saw and do it. So, oh my, okay, I better rush along. So what we found then is that we had the ferret soft palate that has all the soft palate has 2 surfaces. There's a nasopharyngeal side, submucosal glands, and the oral cavity side. And it turns out that the nasopharyngeal side of the soft palate in ferrets, pigs, and humans is just a lawn of alpha-2/6 long chain receptors. So that's where the virus mutated. When it bound to that, it mutated. So this is my lab at the NIH. We started small and grew into a larger group. Everything that I did there was very translational. So then the next fork in the road was the move to Australia, and I was headhunted for this job. Decided to take it up because it would give me a chance to bring my public health training and ID training and everything, and also move up to sort of flu at the global scale. So I'm at the Doherty Institute, which is in Melbourne, and I'm the director of a WHO Collaborating Center for Influenza. So this is, there are 5 such centers around the world: CDC, London, Beijing, Tokyo, and Melbourne. Melbourne's had a WHOCC since 1992, and I am the 4th director of it. And we monitor influenza viruses and do virologic surveillance from around the world. And our main task is to recommend what strains should go into the vaccine twice a year, once for the Southern Hemisphere and once for the Northern Hemisphere. So our Southern Hemisphere meeting's coming up in September. I also have a research lab in the University of Melbourne in the Department of Microimmunology. During COVID, the research lab pivoted completely to COVID, almost completely. And we've done 4 things that I think are still very influenced by my interest in translational medicine. So early on during COVID, as you remember, we didn't have cases. And I had worked on SARS-1 and MERS. So I was hot to trot, and I knew coronaviruses to some extent. And we had a PC3 lab, so I wasn't afraid of working with it. I'm used to putting on a respirator and doing this. So we worked with a number of stem cell researchers from MCRI and Monash who gave us heart, lung, kidney, and placental cells. We infected them so we could study the biology. We looked at how the virus enters, what the host response is, and we looked at which drugs work and how some drugs work much better in one cell type than the other, and because it's based on how the virus enters these cells, and the toxicity is very different. So we've learned a lot from that. Continue to be very interested in airborne transmission. So we now are working with a group of aerosol biologists, engineers, infection control specialists to study airborne transmission and to see how to model it better. I've set up neutralizing antibody assays and mouse models for SARS-CoV-2, and I also serve on a number of advisory panels for the WHO on vaccine products, for emergency use listing, for human challenge studies, and I chaired the group that recommends what strains should go into the COVID vaccines for now. So what are the best things about postdoctoral training wherever you are? And I call this postdoctoral because it doesn't matter if it's a PhD or postdoc. It's an amazing time. You get intellectual and material resources. It's the best time of your life. Take advantage of it. You have a peer group for life. I have friends that I've had since I was a postdoc, and they will be my friends. They're scattered around the world. There's an opportunity to meet and interact with people with very different scientific and cultural backgrounds. The building blocks, I think you need strong medical and scientific training. You need opportunities. So being at the right place at the right time really matters. There's a lot of luck involved, but you also have to take initiative. So you attend seminars, you realize people use the same tools, similar tools, to answer different questions. So think about how you could apply them to your question. Doing it all. Some people do it all successfully. I have chosen to do translational science in my research career. I no longer practice medicine. Of course, I don't have a license to practice medicine in Australia. I kept a license to practice medicine in the US, but really never did it. So translational research has been my way of doing it. It's very important to have clinician scientists who can bridge the divide. It really takes a village to understand human disease, so we really need to bring our perspective to the questions. So my general advice, remain curious, challenge yourself, keep an open mind, and seize opportunities. Opportunities come your way. It may not always be convenient. It may not always be what you had planned to do. I would say keep a sense of humor, stay grounded. And it's very important to find mentors. They don't have to be in your field, but find mentors. And when you supervise people in your life, try to figure out what motivates them and teach them good scientific habits from early on. So as I said, take advantage of opportunities and try to balance new opportunities with the commitments you made. I think you have to have a body of work at the end. It's not just the splashy, flashy papers. It's a body of work, and so dropping what you committed to do in order to do something new is not a great idea. So try to figure out how to do all of this, recognize your strengths and build on them, and develop collaborations. But it's important because it opens up new ways. I mean, I've never worked with stem cells before. I didn't know anything about building engineering, and I know much more than I did before. But it is important to discuss the credit and authorship ahead of time in collaborations. Don't be afraid of lab research. Medicine is very much a ladder, and as you climb, you drop down and you start again. And I think a lot of physicians get intimidated when they go into a lab and they're being bossed around by an RA who's teaching them how to pipette and asking them to clean up after themselves. But that's just good lab citizenship. Spend the time. I mean, I spent 5 years and absolutely loved it. And keep the big picture question in mind in terms of translational science. Ask yourself, what do we know, what do we not know, and what are we proposing? In fact, one of my friends taught me this. She gave me a book that goes through this argument. And she said she makes every postdoc who meets with her every week to say, okay, start with that, just as a reminder. Publications, you have to persevere and complete your projects and publish them. Unfortunately or fortunately, publications are the currency of our science, and having a body of work may be more important than 1 or 2 high-impact papers. Never walk away from the reviews and refuse to do something. Think about it with a cold, walk away from it for a while. Come back and revisit it. It could improve your work story. And I want to close with a word on leadership, because that was a section. As I said again and again, be open to opportunities and challenges, figure out your strengths, your style, your management style, and recognize that we all have areas for improvement. Figure out what motivates members of your team. Find a mentor or coach. When I moved here, I asked for an executive coach because I was moving to a completely different culture, and I didn't want to be labeled the aggressive American. So even though I was coming in at a senior level and would be making changes, I didn't want them to be dismissed in total. Seek leadership training if it's available. You need good support and a sounding board. And step back from time to time and reflect on what has given you satisfaction. So that's my journey. And I love this one piece, and I will just leave it up here. It's called Desiderata. It was found, actually, in a church in Baltimore. And these are bits that I just think are really good philosophy. So with that, I will stop, and thank you once again for your attention.
Thank you so much. What an insightful talk, especially in today's climate where respiratory viruses seem to be at the forefront of everyone's minds. We'll do a few quick questions just so we can stay on time, and perhaps if there are any other questions people haven't asked, perhaps Professor Subbarao will have some time during the break at lunch. Just one over there.
Thank you, Professor Subbarao, for your talk. It's really inspiring, and it brings up a lot of different thought-provoking ideas of clinical research. I myself am a public health nurse, so community health nurse trained in public health. During my public health training, I did a paper around the EPI challenges in Vietnam. Just wanting to hear your thoughts around what the EPI challenges you might have faced when you were doing your research in the United States around trying to address Streptococcus pneumonia.
What challenges?
Oh, the EPI routine challenges. Sorry.
You mean EPI as in EPI vaccines?
Vaccines.
Oh,
yeah.
okay.
Yes.
Yeah. So EPI is the extended program for immunization that the WHO recommends that all children around the world get. So the US is an unusual place in that even though the WHO makes recommendations for the whole world, the US does what it wants. And they have the luxury of doing it. They don't necessarily do it. Not every citizen of the United States gets equal care, as we saw very much so during the pandemic. But there are challenges with immunization in children. And I think a big reminder is what's happened during the pandemic. A lot of children have not received their standard immunization. So I wasn't involved in the pneumococcal side, but we have talked a lot because the influenza vaccine is now universally recommended for children in the US. So there are differences between what the US does and what the rest of the world does.
So just one final question, and then we'll press on with the interest of time.
Thank you so much for that talk. That was really interesting. My name's Sophie. I'm a first-year medical student at Deakin, and I just had a question about all the forks in the road that you spoke about and what kind of drove your decision-making at each of those points.
Yeah, so that is always complicated. I mean, part of it is just where your interest lies at the time. And so, I have circled back to trying to bring the rest of my training in line. in taking the next position. But I do think that it was important to reflect back afterwards and say, was that fun? So when I worked at the CDC, what the CDC does is enormously important, but it wasn't for me. Because I realized after doing it for a few years, I would go to science meetings and people would be talking about what I used to work on. And in the meantime, I worked on H5, but I also worked on a bunch of duds. And so at the end of it, I said, okay, you know, what they do is very important, just doesn't work for me. So I pulled out of there and went to the NIH. And so at each step, I think you have to take into consideration, I mean, I've been mobile, so I don't mind picking up and moving. Many people are sort of restricted in their mobility because of family, children, property. I would say my advice, and it's very biased, is don't ever let property hold you back. You know, you can find all the other excuses, but a house, really? There are houses everywhere. So, yeah. All right, thank you.
Please join me in thanking Professor Subbarao.